So welcome to the cureCADASIL webinar series. I'm Bert Kosiski, the president of cureCADASIL, and I am here with Jane Gunther, the past president and our science officer, and Vanita Ball, who is also a board of trustees member helping with this webinar. And we're privileged to have our guest speaker tonight, Fanny Elahi, who is going to talk on the power of patient-partnered research in CADASIL. Fanny is the principal investigator of our Chan Zuckerberg Initiative patient-partnered collaborations for rare neurodegenerative diseases. cureCADASIL is a nonprofit organization dedicated to helping patients, families, caregivers, and other supporters touched by CADASIL. We aim to unite patients and the medical community toward the common goal of treatments and ultimately a cure for this rare genetic disease by promoting communication, advocacy, research, and education. Fanny is a physician scientist in the departments of neurology, neuroscience, and pathology at the Icahn School of Medicine at Mount Sinai in New York. Her research interests include examining the link between diseases of the brain's small vessels and neurodegeneration, and modeling disease using blood and cells from affected individuals to make discoveries with therapeutic potential and find a cure for CADASIL. The Elahi Lab at the Icahn School of Medicine at Mount Sinai uses a multidisciplinary approach to model disease, identify early biomarkers, and find therapeutic targets for vascular brain dysfunction and degeneration. They focus on genetic causes of vascular neurodegenerative disease to understand fundamental mechanisms that lead to disease pathologies in order to identify impactful therapies. In November of 2022, cureCADASIL and the Elahi Lab at Mount Sinai were awarded the Chan Zuckerberg Initiative patient-partnered collaborations for rare neurodegenerative disease grant. This grant is designed to bring together patient-led rare disease organizations and research teams for a 4-year research project aimed at advancing our understanding of the fundamentals of science of rare diseases. So before I turn it over to Fanny, I want to just give you this note that the audience will be muted during this presentation, but we definitely will take questions at the end. And so if you have a question, you can type it in the question box that you should see in your drop-down on the right side of your menu. And so without further ado, I'm going to change over to Fanny, who will give her presentation.
Can you see my screen?
Yes.
Okay, perfect. So thanks so much, Bert, Jane, Vanita, and cureCADASIL for inviting me to give this talk. It means really a lot to speak to patients, families, and those affected with CADASIL. I just wish that we were seeing each other. So I'll do my best without seeing your faces and having that sort of human contact. I will do my best to deliver a good talk. So our goal is nothing short of curing CADASIL. As a community of researchers working on this, and my lab is 100% dedicated to this mission. We are realistic. We don't think we're going to have a cure immediately. We think we're going to have treatments that will alleviate pain and slow down progression until we buy time to develop cures. Really, frankly, the monogenic diseases will have genetic engineering cures. There are all sorts of genetic methods for altering DNA mutations that may become available sooner than later. But for diseases like CADASIL, where it's not a developmental disorder, life expectancy is not that short, the bar to taking something in humans and doing definitive treatments, such as, say, CRISPR gene editing, is really quite high. So for the next foreseeable future, we need treatments. And in order to develop really impactful treatments, we need to detect disease before irreversible damage has been done, because we don't have regenerative medicine at our disposal. We need to detect what needs to be targeted with therapeutics. You know, when you investigate disease, you're studying human beings, and there's a lot of variability and a lot of differences between human beings. So we need to determine what is most closely associated with symptomatology in order to put our eggs in that basket and develop treatments for that biology related to the most important symptoms of the disease. And finally, we need to be able to detect at scale. So our detection tools cannot be invasive, they cannot be too expensive, they cannot be onerous, in order to be able to treat with precision at scale. We know that the classical syndrome of CADASIL is rare, but 1 in 400 individuals carries a mutation or a rare variant. Therefore, traits or components, parts of the disease can manifest in a much larger portion of the population. This is incredibly important because we stand the chance to understand disease at a different scale, and also to help a lot more people and grow our community. So I think with large population-based studies, and we have one ongoing at Mount Sinai, our understanding of the connection between mutations and disease will advance, and we will have better numbers as well of how many people are actually affected with CADASIL. Maybe not the classical presentation of CADASIL, but components of CADASIL nonetheless. But we have some serious barriers because CADASIL is affecting the tiniest of the blood vessels of the brain, and we don't currently have any direct measurements of the small blood vessels of the brain. What we can do, however, is be creative and develop methods to study the downstream ramifications and impacts of small vessel disease on the microenvironment, on the environment surrounding the blood vessels. And finally, I think one of the biggest barriers has already been lifted. Many of many of individuals affected with CADASIL that I see in clinic come to me because they've had genetic testing because a parent or a family member was diagnosed symptomatically, but all they have is the knowledge that they carry a mutation and they're not symptomatic. That's an incredibly powerful opportunity to intervene at stages of disease that we did not have in the past. Another entry point or another route through which people get a diagnosis is that they can have migraines and get a brain MRI. And based on the findings of the MRI that would be silent from a clinical perspective, they get a diagnosis. All that within preclinical or very early stages of disease. What my lab does is we want to understand what are the pathologies, the molecular pathologies that are connected with the clinical symptomatology or the earliest abnormalities, either on imaging or in symptoms. So I'll give you an example. You know, if the thinking process is slowed, we are even now considering that we could potentially find better ways to quantify migraines and headaches, and maybe those would be the symptoms that we would connect the molecular markers to, because we don't want to understand the biology at much later stages of disease. I think that's important, but the treatment targets may actually differ based on what stage of disease people are at. And then the incredible opportunity for a disease like CADASIL, where there's one gene that's mutated leading to disease, is that we can derive stem cells from individual patients and model disease in vitro, in a dish, in the lab. Why this matters is that when we do the modeling using blood samples and look at thousands of molecules and identify what's abnormal in the blood of individuals with CADASIL and what that means for the pathology, it's all correlative. What that means is that things are linked together, but what we don't know is whether they're driving disease or whether they're causal. And that is actually very important if we're going after something therapeutically. So the term biomarker may be something that you've heard. Biomarker means biological measures or markers of a disease process. It doesn't mean that they're causal. They are marking disease. So we want to go beyond biomarkers. Biomarkers are incredibly important so that we can do better prognostication, so that we can do better risk stratification, but ultimately we need to ensure that the molecular pathologies are within the causal pathway, and that's why we do this in vitro modeling. There are also alternative methods using various kinds of animal models, and in collaboration with companies and other labs, we are exploring what would be good animal models, not mice, not rats necessarily, but more creative models that would allow us to, say, visualize blood vessels and such. And ultimately, all this effort is in order to be more precise with regards to understanding of disease in each individual, because there's, as you well know, there's a huge amount of variability in CADASIL. So I'll show you some very, very technical results, but I want to share this with you, and I'll go through it slowly. And at the end, feel free to bring me back to this and have me explain more if anything remains unclear, because I want you to see what incredible opportunity we have now that we didn't have just 5 years ago, and what this really means for our understanding of CADASIL on a molecular level. So what we do is we collect blood, and thank you to all of you who have donated blood samples. And we do a lot with the blood that you donate. We create stem cells, as I mentioned. We look at thousands of proteins in the blood. We isolate immune cells from the blood and study them to understand whether there's a signature and what's abnormal about the immune system, if anything. And through the generous donation of brain when individuals pass away, we are also studying at a more accurate level, but at a later stage in life, the same pathologies that we detect in blood. And interestingly enough, so far, we're in the early stages of brain tissue analysis, but what we see is that there's some things that we detect really early in the disease process that are no longer there. And as disease progresses, there are new things that emerge that we can see on the brain tissue. So our initial gut feeling that disease evolves, and that means that what we would need to go after early in the disease, mid-disease, or late disease may be different, is starting to substantiate as we look at blood and brain tissue. Both of these are incredibly important to the research. And then the third level is to take the cells from blood, derived stem cells, differentiate them. So you basically deprogram the cells to make them pluripotent. And then through molecular reprogramming, make whatever cell type you wish. And what we're doing is we're building blood vessels and brain cells, various kinds of brain cells, to look at the pathology in a dish. I'll speak later about the incredible collaborations that were established thanks to cureCADASIL, bringing groups of researchers together to take this kind of work to a whole new level. I should add that it's since 2011 that we've been able to do anything with stem cells, and now where we're at is that we're able to build blood vessels in a dish, which is really incredibly powerful. And ultimately, the reason why we're doing this is not just curiosity driven. And in fact, when you work on a disease, it becomes a mission-driven project. And I'm a physician scientist, so I see patients and I have that motivation. But I would say that the PhDs in my lab are also working with the same sense of urgency and at each turn we're asking, if we follow that, is that going to be therapeutically relevant? Because ultimately what we want to do is to use these models and screen drugs, various kinds of drugs in vitro and various kinds of intervention in vitro before we take it into human beings. So at this point, we've looked in each sample donated at 7,500 proteins or thereabouts, and also quantified very specific molecules that we hypothesized to be abnormal. And what we do with that is that we apply lots of different analytical methods, and I'm putting 3 of the many faces in the lab who are working on analytical pipelines. There were the first 3 who joined and did a lot of heavy lifting to establish this, these analytical pipelines in the lab. And I won't get in the detail of it. It'll be too nitty gritty, and I don't think you need to know the details to understand the promise. With machine learning and artificial intelligence, we are able to ask in an unbiased fashion without knowing what would be abnormal, what is abnormal in the data, and that's very powerful. We are also looking at what could potentially be molecular markers, and that has its own kind of analysis. And finally, we are seeing whether just by looking at someone's blood, nothing else, we know nothing about their mutation, we know nothing about their gender, age, symptomatology, are we able to classify individuals into various kinds of group? And why we do that is because we want to see whether the signature in blood is strong enough. And what that means is that we would be able to detect just from blood disease stage, and that would be really incredible. So I'll show you just one slide on the machine learning approach, and here I'm showing individuals with very mild, I would say maybe almost pre-symptomatic, they're barely symptomatic, maybe they just carrying the mutation, individuals that are moderate to more advanced disease, and controls. And by applying machine learning methods, we're able to cluster these individuals in various kinds of groups. If we take the set of proteins that allow us to classify individuals in these groups and ask what brain cells, what vascular brain cells are expressing this circle here, up top, you have different kinds of cells, components of brain's blood vessels, and down here you have a set of proteins, and the links between the set down here and the set up there are showing you which cells are expressing which protein. And what I want to draw your attention to is that the pattern that we detect in early disease, we call it early disease signature, is very different from the pattern that we detect later in the disease. This is just to show that we have 2 papers out on this. And this is not because we're excited to get papers, we're getting other kinds of papers out, but this is to show how far the research in CADASIL is coming. We are one of the many groups publishing on CADASIL, and what's really wonderful is that some things that we find are also found in different cohorts, on brain tissue if we found it in blood, and vice versa. And so the science is rapidly evolving, and we're becoming more confident in the findings that we have. In this paper, we found that there is a signature of changes to the plasticity of the blood vessels. There are changes to metabolic function of the cells, and a term called fibrosis, which means loss of functions of the cells, and think of it as sort of scar tissue formation. And in this other paper, we found that the signal from inflammatory signatures are associated with various kinds of brain imaging findings and also cognitive slowing. So I mentioned that we do this associative correlative work in blood, but it's blood. We don't know the pathologies that we're detecting, where are the signatures coming from, and ultimately we need the level of precision that we can get only with tissue. Now, if we go to brain tissue, it's end stage. So what do? We take the human blood cells, we turn them into stem cells, and then we make various kinds of primary cells from them. And this is work from a talented postdoc in the lab, Stephen Fitzsimons. And here what I'm showing you is one kind of cell type in blue, another kind in red. The same cell types are now colored in yellow and red. And here you see a combination. This is basically blood vessels formed in a dish. And here I'm showing you a video. So cells are programmed for specific functions, and you can recreate that in a dish. Why this matters is that this is human tissue. This is tissue from someone who would have a certain mutation and be affected with CADASIL. What we do is that we create controls for that. So we know what's artifact and what's driven by the mutation. And once we get a clear abnormality in a dish, that's when we get to screen the drugs. And we're on our way to getting a clear abnormality in a dish that we're really excited about. This is again work done by Stephen Fitzsimons. And here he looked at the formation of these networks and found that the formation of the networks was slightly different in cells carrying a mutation in Notch3 than controls. In collaboration, this is one of the powerful collaborations that cureCADASIL brought together. So in the framework of our Chan Zuckerberg initiative, we were connected with the Shrek Zhang's lab at Brigham, and he's a bioengineer. So he is building these tiny little, he's 3D printing tiny little molds for us that we use to seed the cells. It's basically creating a blood vessel lumen. And why this is important is that what I just showed you in a prior slide is cells that we cannot flow blood through. And to study blood vessels, one of the biggest obstacles and hurdles in the field has been the lack of blood flow. And so for the first time, thanks to this collaboration, we're able to have blood flow. And here you can see a little bit of ink going through a lumen to show that there is flow. This is another figure showing that this is basically a blood vessel, and these are all sorts of other cell types around the blood vessel. And so we're recreating microstructures in the dish. Now, this was the molecular component, and it's incredibly important if we are to develop good biomarkers and therapeutic strategies to help alleviate the burden of disease. But why we're doing this is not because we're in love with molecules or absolutely curious. Our curiosity could be driven to study something else. Why we're doing this is that we are touched by the stories, we feel part of the community, and one realization that we had is that we think we know CADASIL, but do we really? What bothers people? Why is CADASIL disrupting lives? We want to know what problem people would like to have solved. We think about cognitive impairment, memory impairment, ultimately the possibility of dementia, but what if it's mood fluctuations? What if it's headaches? What if it's fatigue? What if it's temperature dysregulations? We have serious blind spots. And so the grant that Birch just mentioned, the Chan Zuckerberg initiative, was an incredible opportunity to partner with cureCADASIL, and through cureCADASIL, build a patient network that informs the research team. Again, I see a lot of patients in clinic, but I wanted my scientific collaborators and people in my lab to get the same emotional attachment to the disease and the same sort of mission-driven purpose to solve problems and help individuals with CADASIL. So this is the scientific team. These are the principal investigators. Many people in each lab are working on CADASIL. Joel Blanchard is an expert in stem cell modeling. Tawfiq Raj is a computational biologist, and Shrik Zhang, you already heard, is a bioengineer who's building unique models for studying blood vessels in a dish. And our partnership with cureCADASIL has been powerful. We meet regularly, we think together, we disagree with each other, we agree with each other. We've really built a close relationship, and I'm just really impressed with all that cureCADASIL is doing for this disease. And they have their own aims, which is building a stronger community, advancing advocacy, education, and continuing to bring scientists and clinicians together. So this was the goal of our project. What we are after is to discover clinically relevant, again, things that actually matter to people, and the molecular underpinnings of these clinically relevant symptoms, and understand fundamental mechanisms that we can then go after therapeutically with drugs and various kinds of interventions. In doing so, we came up with several questions. One, identify the drivers, not just correlates, but drivers of disease. What is actually advancing the symptoms that bother patients most? And then, because we had some interesting findings with the proteomic approaches, when I mentioned the 7,500 proteins, we were really intrigued by the function of the peripheral immune system. And this is why we're really grateful that some of you are flying to New York to donate blood, because we can only do this with fresh blood collected. So we're extracting the immune cells and understanding what could potentially be abnormal about the immune system in individuals with CADASIL. Reason being that it's incredibly important from a therapeutic perspective. We can reach the immune system much better than brain cells, and there are all sorts of various tools, therapeutic tools that are being developed to modulate the immune system in more severe diseases. So this would be an incredible therapeutic opportunity. And our biggest challenge is that we are modeling brain tissue, and brain is the least accessible tissue. So we have to wait until end stage and donation, generous donation of brain tissue when people pass away. So what do? We are going to stem cells, but stem cells have some serious limitations. So one of them being absence of flow. So we're trying to basically solve that. And I'm incredibly impressed with the progress that has been made with this multi-team, large group or team, how synergistically people are working together, and how very little ego I've seen in any of the interactions. It's been really an honor and pleasure to work with this team of investigators. I'll skip this, but the one point that I want to, so these were the aims of cureCADASIL in this collaboration, and I want to just draw your attention to the community advisory group that has been such a powerful engine of progress in this grant. This is the cohort that we've been able to build in partnership with cureCADASIL, with you, with researchers, clinicians. I was really touched by how willing the community affected by CADASIL is to participate and contribute to advancement of knowledge in CADASIL. And so we have a local US-wide cohort where people are willing to fly all the way to New York. Recently, we've been able to start a fully remote cohort, meaning that we can send mobile phlebotomy to people's homes, and that has really helped us expand this cohort. But look, I mean, there are individuals across the globe that also want to participate, and we want to build the community. So even though we can't collect blood, and oftentimes there's a language barrier, we want to stay connected to the community, and we don't turn anyone away. This is the incredible community advisory group, and I've learned so much by interacting with this wonderful group, and they've really guided and focused us on important matters. And in collaboration with this incredible group, and really spearheaded strongly by Sheila Connor and Andrea Barroso, they launched a patient survey some time ago, now a few months ago, and we froze this in March of this year. And I'm going to show you just a teaser, because if you want to see the full scope of results, which are incredibly interesting, please attend the patient investigator meeting on the 29th of June. So the participants came from all over the globe, a majority from the US, but really from all over the globe. And here, truly, I think the barrier was that we only send the survey in English. We would love to translate the survey in other languages. And through this incredible community advisory group, there are researchers in Japan and hopefully Korea and elsewhere who are translating the survey to circulate it in native languages. So the teasers are, I'm just showing you the age distribution. Not surprising, this is sort of what we're seeing. I think maybe it has to do also with how we distributed the survey, but this is sort of a normal looking age distribution. As always, we have more women participating than men. This is also congruent with the distribution of our research cohort. By far, the most prominent symptom that prompts a diagnosis is migraine. Second is TIA and strokes, and then family history. And this is what I was talking about, which is absolutely incredibly wonderful. Now, CADASIL is getting recognized. I think when I ask a lot of questions when I meet patients, and I always want to know who was the first person in their family who got diagnosed. And oftentimes there's stories, but no one ever reached a diagnosis in grandma or grandpa. And it's really their immediate parents or uncle that would have gotten a diagnosis. And now we can do genetic screening. So that individual at a very early or completely asymptomatic stage of disease is receiving a diagnosis, which I think is truly a game changer, because there's a lot that lifestyle can do for the course of disease. And then much less frequently are the other symptoms, including cognitive impairment. So this is the QR code. I'm going to leave it on for a few seconds, and I'm sure it can be distributed again. If you haven't taken the survey and you're interested to contribute data to the survey, please scan this and do so. And maybe cureCADASIL can recirculate the survey so that you can participate. Ultimately, there are many, many different components of abnormality that can be noted when we look at a human being with a mutation, without a mutation, with disease. We have to bear in mind that we're thinking about a mutation in a specific gene, but the rest of the genome is contributing to that person's medical comorbidities, lifestyle, experience, symptoms. And so there are many degrees of freedom. And what we want to do with the molecular measures in combination with other biomarkers that others are developing, such as imaging, retinal biomarkers, which we looked at some time ago, and I know others are now looking at, all these in combination could potentially do this, to pinpoint with much greater precision where we should be looking and what we should be targeting. And I'm going to add in whom, because I think the targets are going to be different in their impact depending on what stage of disease an individual is in. And I just wanted to show you the faces of all the young people in the lab who now know about CADASIL, who are now part of your team, completely rooting for you and working with passion and a sense of urgency to advancing knowledge towards development of better treatments. This is not even all encompassing. There are 4 additional individuals who have joined the lab who I don't even have a photo for. So, you know, often I speak to patients and I hear not infrequently that their physicians or their or individuals in the care system, in their local care system, don't know about CADASIL. Well, this young man who was a medical student when he worked with me is now going to be an emergency medicine resident. So he will know about CADASIL. He will be a pathologist. Many scientists and many younger individuals at earlier stages of their training who don't even know what they're going to go into, but maybe science, medicine, or the combination of the two will know about CADASIL. And I think this will have a crescendo effect and more people will learn about CADASIL who could potentially do something about CADASIL. I want to thank, you saw their faces, at least some of them, really motivated, hardworking, passionate individuals in the lab who are working on CADASIL, incredibly powerful collaborators, cureCADASIL being one of them, you being on top of the list as participants in research, and the funding and sponsorship that we've received from various organizations, the most important one being Zuckerberg, which has not only funded our research, but has also connected us with the larger community working on CADASIL. Thank you so much for your attention.
So Fanny, this is great. And we have a number of questions, and I think probably more will develop as people get into this. And I'll just go down the list here. One was a question about vitamins, and there were several parts to this question, but just in general, are vitamins potentially effective? Do they cross the blood-brain barrier? So there was one of the participants was interested in that particular issue. If you have comments about vitamin therapies.
Absolutely. I'm trying to stop sharing my screen so that I can see faces. How do I stop sharing the screen?
You hit.
Oh, I can do it. Okay, perfect. No. Okay. I can see you now. Okay. The vitamins. It's a really good question. So at the patient investigator meeting, we're going to reveal the rest of the survey, and we asked specifically what people are taking, including vitamins and other things. And we don't know what vitamins may be impacting CADASIL disease pathology specifically, but we know some things that are good for the brain. And so I always try to make sure that my patients are optimized from all other perspectives, one of them being B12. The threshold that is typically considered low is way low. So you want to be around 500 or above. The other one being vitamin D. It's incredibly important for the immune system. So making sure that you don't have low levels of vitamin D. And then L-arginine and other things I think people are taking, there's no harm. And it's a matter of trying. But I have to say one of the motivations for building this screening platform is also to try vitamins. Basically any molecule could be tested to see whether it's going to impact anything at all. Short of that, we would need human clinical trials to have definitive answers as to what combination of vitamins and what dose would help. But I'm hoping that we can de-risk and make things cheaper and not do as many clinical trials by testing things in vitro in a dish.
There's another question. What kinds of immune regulation issues are you seeing in your data? I ask because I have been on IVIG for primary immune deficiency, and it has helped my quality of life.
That's an interesting question. So we're seeing signals from various immune cells being activated or dysfunctional. This is sort of smoke, not the fire, because these are molecules that we're detecting in blood. A first pass with a very limited technique that we did on the blood immune cells, we see very, very, very small differences to the point where I can't say that they're substantial abnormalities if you just look at the quiescent immune system in CADASIL, which is not so surprising because if there were really loud abnormalities, it would probably be an immune disease. It would be classified as an immune disease. But what we're doing next is we're stimulating the cells, because in many of the diseases that are not classical immune diseases, it's the response of the immune system that's abnormal. It's not necessarily the numbers of a subset of immune cells, which is all that we sort of looked at. So we'll know more. I'm taking the molecular signatures seriously, and I think there is something not normal about the immune system, but through these additional experiments, we'll know. And the other thing that I should add is that there's a little bit of a chicken or egg, because blood vessels can affect the immune system and vice versa. So we also want to test to see whether the immune system's signature is just a response to abnormalities of the blood vessels or not.
There's a couple questions here that bring up the issue of awareness of the disease out in the general medical community. This one, before 1993, it was named, quote, lacunar vascular disease, end quote. And my father-in-law, his father, my husband's grandfather were institutionalized as crazy, but then subsequently found to have CADASIL.
What's the question, Bert?
Well, that's the comment. I have a question, though, that relates to getting information from patients, and particularly in rare disease. I mean, it's important for all diseases, and the FDA has put a lot of emphasis in this on drug approvals more recently. But it's particularly important, I think, in rare diseases where issues are different. And so, how do we go about finding patient recorded outcome measures? And the key word here is measures, because the FDA wants, they definitely want input from patients, but they want something that is measurable and reproducible. And I think that's the challenge. And the survey, I think, is a great place to start, but maybe there are things that could be done to refine this. Are there specific measures that is specific to CADASIL and not necessarily other diseases that may ultimately prove to be an important outcome should someday, hopefully, we'll have a therapy to test in clinical trials.
Yeah, so just want to add something to the comment that was shared. I think that's a really powerful story, and I think we need to get more stories out there. So the title of the last section was From Molecules to Stories, because that's how we're going to expand the community. And this was the story for individuals suffering from frontotemporal degeneration as well. It's psychiatric presentation. So the comment is actually linked to the question that you have, Bert. I think we've gone for the easy quantification. It's easy to quantify cognitive impairment. We administer these tests and people are unable to do them as well, and we quantify that. It was by far not the most important symptom for the patients or for the families. And so things that are much harder, we've sort of pushed under the rug, but we can't do that because the reality of most diseases is actually the psychiatric component is the most bothersome. And I think we can borrow from the other disorders that have had to deal with the psychiatric symptomatology. And as much as I hate long COVID because it's so hard, I was part of a long COVID clinic for a little bit, and it's really hard to dissociate what was pre-long COVID and what's post-long COVID. But there are also really good measures being developed for fatigue, for brain fog, and for fluctuations. And I think these are actually things that I hear patients complain about. Fatigue is one of the biggest problems that patients face. But the psychiatric symptomatology, I think, is still a big gap. I don't think we have really nice quantitative measures, but we need them. And so some of the things, some of the very creative measures would be passive collection of iPhone usage. Sorry. Ignore the sound in the background. Hopefully it's not very loud. But it's iPhone usage or habits or internet browsing information. You know, there may be really creative indirect measures that we can gather. I know that some pharmaceutical companies are working on social media. So how do we know that people are feeling better or worse? Can we gather that information from their social media posts? Yes, Mom. Okay, okay. Real life.
Yes.
So
here's another related question that has popped up, I think, as a result of this discussion. Do you know why there are so many variables in how the disease expresses itself in different individuals, including within specific families and siblings? In other words, is there an explanation for the heterogeneity of this disease, which is notorious?
yes, it gives me great hope. I love that it's a very heterogeneous disease because it means that there's only so much that is determined by the mutation, and that there's a lot that we could do to modulate the course of disease. Part of it is probably genetics because those family members are sharing part of their genome, not the entirety of their genome. But also, you know, we are the expression of gene interacting with environment, and environments are not completely identical. So what I didn't mention in the talk is that we've looked at the genetic data in our Mount Sinai health system, and we found about 180 individuals who carry a mutation, but many of them don't have any record in their medical chart of ever having presented with a symptom or having received a formal diagnosis. So I think the 1 in 400 individuals have a rare variant or a mutation in the UK Biobank is also what we found. And that means that you could actually be asymptomatic and have a mutation in Notch3. And that speaks to other kind of biology, the resilience biology that we are also interested in understanding. I think we need much higher numbers in order to be powered to understand what could protect individuals against the mutation.
Is there a specific question, and it's one I know we're working on, is who would I contact to donate my brain at the time of death for this research?
So Bert, very specifically in cureCADASIL, has partnered with us on creating a smooth pathway. I think until we have something in place, just email me, copy cureCADASIL, because we are coming up with solutions depending on where you are. Eventually, we want to have a very clear system where you would contact your local team, they would accept the donation and they would ship it to us. But until we have that in place, we just work on putting it in place in time to accept the donation. Brain tissue is incredibly important, and thanks so much for considering that. So we'll do everything in our power to make it happen.
Another heterogeneity question. Exons 1 to 6 have been labeled as more severe form of CADASIL, but some people diagnosed with 1 to 6 seem to be doing well. How is that explained?
That's absolutely. I think this goes back to, we really need to expand the cohorts and the population, because we don't see that in our cohort. It was true in one study, and I think there are factors that we don't necessarily understand. My dear colleague and friend, Michael Wang, who I think gave a talk to, gave a seminar similar to this, is doing really beautiful work to understand how the interaction of the various mutated proteins is impacted by the location of the mutation. And I think that's going to be really illuminating. And we are doing similar work where we're taking stem cell lines that are identical from every perspective except for the location of the mutation, to see whether this is recapitulated at that kind of accuracy level. I reassure my patients, because we're not seeing that in our research cohort. So when you get your result back and you have a mutation in the repeat number 4, I wouldn't necessarily think that you would have a more severe phenotype.
Another question. I have been diagnosed with HTRA1. How closely connected do you think this diagnosis is to CADASIL? And do you think that future therapeutic developments may also help?
I do think some of the biology that we're uncovering is getting connected to HTRA1. There's some incredible results also coming from other kinds of vascular conditions leading to HTRA1. So I think I think as we advance our understanding of why the blood vessels are getting affected, we're also going to shed light on how HTRA1 is implicated. And if you wanted to participate in research, you're absolutely welcome to participate in our research, because we're not limiting our research to Notch3 mutations. We actually think it's really important to expand the scope, and in order to understand specific and shared biology.
Great. A couple of comments here, a couple of thank yous. Thank you, cureCADASIL and Dr. Elahi. And also a question, are these slides available for download? I know that we will certainly have the recording available. That should become available in a day or two. So if you want to listen to this recording, or if others who registered want to listen to the recording, that will be available. I don't know about slides.
I did not spend enough time on the slides to make sure they're perfect enough for them. If you're interested in any specific data, I would be more than happy to share. Just email me and I can share slides with you, absolutely. And if any questions come up, I always tell everyone my door is open. The electronic door is also open. I'm always happy to speak with you and help in any way if I can.
Well, this has been fantastic. Fanny, thank you very much.
Thank you.
I'm going to switch to my screen if I can here. And I'm going to thank also all of the participants. It was a good turnout, and as usual, I'm sure more will be watching this recording as time goes on. But it was a great webinar. We appreciate all the support we get with cureCADASIL. We are moving forward. I'm very hopeful that research like Fanny is doing is really going to yield fruit in the future. So I think on that note, I will end it. And again, thank you, Fanny, and we'll be hearing more from you, I'm sure. Thank you.
Thanks so much. Bye-bye, everyone.
Anyway.